Hello everyone, so welcome back to the latest lecture session. So within the context of, I believe, remediation of soils and sediments, we were looking at soil vapor extraction, right? As an, you have a relatively volatile compound, we are looking at contamination of the relevant gas, let's say, in the Vadose zone, and how do we treat that particular contaminated gaseous phase and so on, right? And then we started looking at bioremediation now, right? So that's something we are going to continue with today. And maybe once we wrap up bioremediation, we are going to look at the relevant case studies for soil vapor extraction and bioremediation, let's see, right? So let's look at what we have discussed thus far. So we looked at or introduced ourselves to a few terms. One was bioremediation, right? So obviously, what do we have out here? Let's say, here let's say the aspects were that we have living organisms, could be microbes or plants, and they bring about, let's say, either degradation or immobilization of the relevant contaminant, right? So if it was only living plants that we are going to talk about or consider, that would have been, or that would be referred to as phytoremediation, let's say, right? Phytoremediation. But when we talk about bioremediation, typically let's say it encompasses both the action by the microorganisms or the relevant plants, let's say, right? And again, the key aspect is either degradation or even immobilization, let's say, is considered bioremediation, let's say, right? And I believe then we looked at other aspects, as in what is biodegradation, let's say, right? So I think we more or less, I mean, it's self-explanatory, right? You have transformation of a relevant compound or a conversion of a relevant compound from one form to the other, and obviously you have biological process involved. So biologically mediated conversion of a relevant compound, let's say, that we refer to as biodegradation, right? In that context, obviously we have mineralization. So when you have complete degradation, let's say, right, towards the relevant end products, let's say, right, you are going to, for example, to CO2 and H2O and so on, let's say, right, then you say that, mineralization has occurred. Typically, let's say, this is what you would want, let's say, because some of the byproducts during degradation could also be toxic, let's say, right. So again, mineralization is something that you would typically prefer, let's say, right, and this is something we, these are some of the aspects we looked at. And then we saw the cases for application of what do we say, bioremediation ex situ and in situ, let's say, right. And ex situ we also looked at land farming or such land piles, let's say, right. But in that context we saw that you need to have a impermeable or semi-impermeable layer beneath, right, because you do not want the relevant contaminant to reach the relevant groundwater or contaminate the soil beneath it, right. So there are different aspects and we briefly discussed them. And obvious advantages were that, let's say, the costs are going to be less, typically relatively more acceptable to the relevant communities, right? Biological process typically more acceptable, typically anyway, obviously, right? And what else do we have? And here typically we actually have degradation, or most of the time we have degradation of the relevant compounds, most of the time anyway. And then obvious disadvantages, time, right? They are typically slow. And depending upon the varying site conditions or site conditions, let's say, or the heterogeneity, the relevant process can be inhibited, let's say, right. You can have some toxic compounds being produced as byproducts. That's certainly an issue, right. So these are some of the major disadvantages, right. Again, we just looked at an overview, let's say, right. So we are going to obviously look at the relevant aspects or other relevant aspects, as in what do I need, let's say, to be able to promote or have bioremediation to occur, let's say, right. So obviously the first aspect would be, right, to have suitable microorganisms, right. So in this context, we also looked at some aspects or similar aspects in the context of natural attenuation. Obviously you want microorganisms, right, but what role do these microorganisms typically play now, right, and why do they play that role, let's say, or why do they want to play that role? So here you have different compounds, and while degrading these compounds, energy will be released, let's say, or while the redox process take place, let's say, typically energy is released, and the microbes, let's say, for either their growth, let's say, right, or cell synthesis, they want this energy, let's say, right. So for that particular case, these microbes play a role of, let's say, catalyst. How do they do that? They release enzymes which act as catalysts, thus shortening the relevant time required, or fastening the kinetics of degradation of the relevant compounds, let's say, right. So obviously you need suitable microorganisms now, right. So what are the different sources of organisms, let's say, right. So there are two aspects obviously, one is natural selection based on natural selection, as in let us say everywhere you are going to have typically microbes present unless the conditions are remarkably toxic to almost all kinds of microbes, but that will rarely be or ever be the case. Let us see, typically you will have some kind of microbes or the other out there, right. So when I refer to natural selection or such, what does that mean now? So based on the relevant conditions at the site, let us say certain microbes are thriving. What does that mean? Let us say they are the fittest, right, fittest for those conditions now, right. Typically we know that we refer to the term, let us say, or talk about term survival of the fittest now, right. So natural selection, so based on those conditions, let us say, or the stress there or extreme conditions out there in that particular site, certain microbes are thriving, and typically those microbes are obviously relatively better off at taking the process through, right. So ideally you would want to promote growth of those kinds of microbes, let us say, and those obviously are from natural selection now, right. So that is one particular case. Or you can introduce microbes, let us say, right, and in that context we refer to that as bio-augmentation, right. So you are augmenting the process, let us say, right, and you are typically going to introduce microbes, and typically these particular supplements, if I can call them, maybe not supplements, these what do we say, non-native species, if I can call them in this particular case, let us say, right, are typically sold, but again they are obviously costly, right, and also keep in mind that they are not, what do we say, very site specific to those conditions. Yes, so obviously you are going to have different variables at play there, right. But typically when would this particular bio-augmentation work? It would typically work, let us say, when, let us say, you have relatively non-biodegradable compound, a relatively non-biodegradable compound in a mixture of relatively more biodegradable compounds, let us say, right. So typically in these conditions, let us say, you will typically have, what do you say, greater chances of success for this particular bio-augmentation now, right. Again two sources of microorganisms, one the natural, let us say, the one that are thriving, or you can introduce non-native species, let us say, from other locations that you have what do we say, cultured and grown and so on, let us say, right, and different aspects out there, right, when we add microbes though, right. So that is something to keep in mind. And how do we go about the looking at the relevant measurements now, right. So measurements, we can look at different aspects. One of them would be directly measuring the microorganisms, let us say, right. So that is one particular case, or measuring the activity that we see or we assume to see, right, or indicators of, let us say, microbial activity, let us say, right. So we are going to look at these three aspects. So with respect to measuring microorganisms, I will just give a very generic overview, let us say, right. So you extract them from the relevant soil, let us say, or dilute them, let us say, right, and dilute them, right. You extract the relevant microbes from the soil or your particular matrix and you dilute them, let us say, and then you are going to grow them, let us say, in cultures, let us say, right. You are going to put in conditions that promote the growth of these particular microbes, let us say, right. So you can do that on membrane, on plate, or in tubes, let us say, right. So once you grow these particular what do we say, microorganisms, you are going to have to count them, right, or there are different methods now, I believe, right, and in that manner you can measure the relevant number of microbes present, as in the rate at which that they can typically grow, let us say, and you can thus estimate the relevant amounts, let us say, right. Again that is one particular case, let us say, where how you can measure the microbes now, right, or you can obviously measure the activity now, right. So activity in the sense, let us say, as we looked at in different cases, you need not directly measure the relevant compound, but you can look at the relevant byproducts or so on now, right. So what are they, or how do we go about that? Let us see. In this case they can be carbon dioxide, let us say, if typically you have your relevant redox process going through, right. What is going to happen? You are going to have, if there is mineralization, the carbon, let us say, is going to be oxidized completely, let us say, I am assuming a particular example. So what would happen in that case? Typically you would estimate or assume that carbon dioxide is going to be produced. So you can look for either effect on pH, or let us say the carbon dioxide concentrations and so on, right. Or depending upon the type of process, you can look at consumption of or decrease in oxygen, let us say, right. You can look at the stoichiometry, right, how much oxygen is required for a particular process to go through, and you can look at the rate at which or the level of decrease in oxygen, and then estimate the relevant activity, let us say, right. So different ways obviously out there, or there are other indicators again, right. And for example, let us say, what are we looking at here? For example, here microbes have the protein content there. So you can look at the nitrogen content, right, to be able to indirectly estimate the amount of the microbial activity, let us say, right. The nitrogen content or the ATP, let us say, can be analyzed for, I believe, right. These are energy carriers here, right. So there are other indicators obviously that you can use to be able to measure the relevant activity at your particular site, let us say, right. So these are some of the aspects to keep in mind in the context of measurement now, right. So let us look at the other aspects. So first, obviously you need the organisms, suitable organisms, so that is something we looked at. Sources and measurement of these organisms, we looked at that, let us see. So now let us say, moving on to the relevant aspect, you need an energy source, let us say, right. So in the context of energy source, obviously there are two aspects to look at. One is obviously that of respiration. So you have an electron donor and an electron acceptor, right. So in the context of respiration, you need an electron donor and an electron acceptor, and then there is going to be the redox process going through, right, and then you are going to have the relevant byproducts, right. This is the redox process, right, reduction oxidation, and then you are going to, or the microbes are going to facilitate this process by releasing the relevant enzymes and thus gaining the relevant energy, let us say, for their own needs now, right. So obviously an example can be let A, which was in its more reduced form, plus B, which was in its more oxidized form, is now transformed into A in its more oxidized form plus B in its more reduced form. So A, which was relatively reduced, now transformed into a more oxidized state, let us say, right. So what is happening out there, or B, which was relatively more oxidized, let us say, is transforming into a relatively more reduced state, let us say, right. So what is happening out there, oxidized form to reduce, right. So oxidized state is decreasing, or you can understand that it is taking in electrons, right. So this is taking in or accepting electrons and transforming into a more reduced form. And what is happening to this particular compound, obviously if this particular compound is accepting electrons, this particular compound is donating the electrons and transforming into it a more oxidized form, let us say, right. So again, one of your particular contaminants will either be A or B, right, depending upon your type of conditions out there, right. Your contaminated compound, let us say, can either be a relatively more reduced compound or oxidized compound, right, and then you can look at promoting which conditions and so on and so forth. But obviously in respiration, as we discussed, we need to have electron donor and electron acceptor and so on, independent electron acceptors and donors. So how does it differ from fermentation though, right. So in fermentation you have one particular relatively semi-oxidized compound, if I may say so, right, and I believe we did look at one particular example or a few examples in the context of natural attenuation, right. So this compound, let us say, will be transformed, let us say, compound X will be transformed, let us say, into a more oxidized form plus more reduced form, let us say, right. So obviously this needs to be semi-oxidized or such, let us say, right. I think we looked at acetic acid or some such example which I am unable to recollect right now, the specific example anyway. So in the case of fermentation, this is what you have a single compound and it is transformed into a more oxidized form and also more reduced form, let us say, right, and that is the case that we looked at. So these are the two cases, and obviously you look at chemotrophs, right. They derive energy from this particular chemical process, let us say, right, or redox process, chemotrophs, let us say, right. So what do we have here? We will have microbes and we have the different or major components for deriving the relevant energy. Is that good enough for the microbes to thrive and degrade the relevant contaminants? Not really, right. So what else is required, let us say, right. So what else now? So in this context, similar to human beings, let us say, you just satisfying one particular need is not good enough. Humans to thrive, let us say, have different needs now. Similarly, microbes have different needs now, right. For example, they need carbon, right. They need carbon now, let us say, again for cell synthesis, and typically let us say your organic compounds, let us say, or let us say heterotrophs, let us say, if I may, relatively more heterotrophic compounds, pardon me, or let me call them organic compounds for now, right. Organic compounds have your carbon, right, and the carbon, the needs of the relevant microbe for carbon, let us say, for cell synthesis, let us say, are met by these organic compounds, let us say, typically, let us say, right. And also what else do they need? They need nitrogen, and also they need a source of phosphorus, let us say, maybe not as high as carbon, but they still need nitrogen and phosphorus at certain levels. So if you do not have some of these key aspects and you just have the electron donor and acceptor, the microbes will not be able to thrive. So obviously all these aspects also need to be looked at, let's say. And then there are other trace compounds that are also required, or trace nutrients, if I may say so. Again, we are not going to that in that detail in this particular class, let's say, because that's slightly out of our particular domain here, let's say, right. Again, what do we need? Rather than just the microbes and electron acceptor and donor, we have other major nutrients and minor compounds or trace nutrients that are required, let's say, right. So that's something to keep in mind. And now obviously you also need conditions, let's say, as in similar to human beings, the microbes also have a certain preferable temperature range, right. So temperature is a key issue. So you are going to have temperature, as in we are done with synthesis needs, we are looking at suitable environment, let's say. So what are some of them, let's say? Obviously temperature is an aspect, yes, there is an optimal range, let's say, typically, and obviously pH, right. Way too acidic or basic, typically the microbes can't withstand these extreme conditions, right. So that's something to keep in mind too, right. So again, temperature, pH, and you have other aspects too, obviously, right. So let me look at some of the other aspects we are going to cover now. So here we are going to look at, let's say, a particular term called bioavailability, let's say, right. As in there are bioavailability actually encompasses quite a few aspects, but I am going to talk about, let's say, the access in this particular context. Let's say I am only going to talk about or restrict myself to, let's say, talking about the availability of a particular what we say compound, let's say, for the microbe to utilize, let's say, right. But again, as I mentioned, bioavailability also looks at or encompasses other aspects which I am not going to look at, let's say. And why is that obviously an issue, let's say, right. And typically bioavailability is affected by the extent to which, or the time to which, let's say, or after which you are trying to kickstart your remediation. We will look at why, let's say. So for example, let's say you have a site contaminated with a relatively hydrophobic compound, let's say, right. So what do we have out here? We have, let's say, a hydrophobic compound out here. This is the surface, and you have the soil particles, let's say, and let's say initially, let's say you have your compound out here, let's say, right, and then it's adsorbed onto the surface, let's say, of this soil particle, right. If this was the initial case and the microbes are present, let's say, they can utilize these particular contaminants too. But what would happen, let's say, over time, and if there were no microbes present initially, let's say. So what would happen now? You would have, what do we say, this particular compound, reaching the relative interiors of the relevant soil particle, let's say, right, or it's no more at the relevant surface now, right. So what does that mean? Let's say for the relevant compound is not available for your particular microbes now, right. So the bioavailability is an issue in this particular context now, right. So let's say if considerable time has passed since the initial spill or the what do we say, contamination of the site, what would happen? The relevant contaminant would travel further into the interior of your relevant soil particle, let's say. Thus, at the surface where the microbes would typically be present or be available, let's say, the compound is not available, as in though within the soil, or if I look at the total concentration, yes, the relevant compound is present, but it's not available to the relevant microbe though, right. So again, let's say there are kinetics at play, but what kinetics now? The rate at which the compound diffuses out of the soil particle onto the surface again, let's say, and these aspects will obviously play a role, right. So obviously bioavailability is a key issue, and let's say any toxicity or such that's affecting the microbes or such are also covered, but I am not going to into that at this particular stage, let's say. So heterogeneity, let's say, right. So why is this important, or how is this important now, right. So heterogeneity, let's say, and there are a number of variables out there in the site, let's say. So typically, let's say if this is my site, again side view, and if it's a homogeneous mixture, let's say, not mixture, matrix, obviously it makes the job much easier for me. But typically, as we know and as we have been witnessing or looking at different case studies, we have never come across a site where the conditions are homogeneous, right. You have different layers, let's say, and so on and so forth, let's say. So how does it affect your particular case? Let's see. Let's look at one particular example, as in, for example, assume we want dissolved oxygen to be around, let's say, 6 milligram per liter, let's say, right. So let's say somebody comes to you and tells you that the average dissolved oxygen concentration is 6.5 milligram per liter. So then they ask you, would you be able to take a call with about is this particular site feasible for bioremediation or not, let us say, assuming that all the other variables are according to order, let us say, right. So again keep in mind, obviously we are talking about heterogeneity here, right. So the key issue is that, let us say, if you had zones where the dissolved oxygen is 0 and zones where, say, dissolved oxygen is relatively high, let us say, right, maybe 10 or 9 depending upon the saturation levels, let us say, and it works out in such a way that the average is around 6 or 6.5, let us say, right. Well, the rates also, or the average rate also be according to this particular DO values? No. Why is that? If it is 0, let us say, for the particular example I am looking at, let us say, which requires dissolved oxygen, there will be no removal or degradation within this particular region where the dissolved oxygen is 0, but there will be removal at that particular region where the dissolved oxygen is available or above the relevant thresholds, let us say, right. But obviously it won't work out as it is an average case, right. I mean, as in you can calculate the average rate and so on, right. Obviously heterogeneity is a huge issue now, right. A huge issue in the sense that you need to have the relevant what do we say, nutrients or the electron donor or the electron acceptor or the dissolved oxygen and so on at comfortable or at the relevant levels, let us say, right. So that is something to keep in mind. And also another aspect is that, let us say, if your site is heterogeneous and you have relevant contaminant being transported, let us say, and here you have a relatively impermeable layer here, let us say, what is going to happen? Your groundwater flow patterns might be relatively different now, right. So again, because of these physical characteristics of the relevant soil or such, let us say, what is going to happen now? You are going to have issues with your particular what do we say, contaminant concentrations, let us say, at those particular localized zones, let us say. So right, so it is again you need to understand that there are different variables at play, and the greater the complexity of the system or the heterogeneity of the system, the greater the complexity of the system, I guess, right. Heterogeneity and complexity of the system go hand in hand now, right. So that is some aspect or one aspect to keep in mind now, right. And obviously the other aspect that we are going to look at is kinetics, let us say, right. Obviously we talked about one of the disadvantages of the relevant what do we say, microbial process based degradation or bioremediation being kinetics, let us say, right. So we obviously need to understand what are some of the factors that affect kinetics here, right. So let us very briefly talk about that and end this session for today, right. So what do you need? Obviously you need the macronutrients, micronutrients and so on and so forth, but obviously you typically need your electron acceptor, let us say, right, or electron donor, let us say. Let us assume that is the case here, let us say, right. As in I have, let us say, assume I have oxygen which is going to be my electron acceptor, let us say, right, and I need an electron donor, let us say, right, and I have some compound X, let us say, right, or S, let us say, right, which is my contaminant which I want to degrade and which can act as an electron donor, let us say, right. So let us say it is some reduced form of carbon, let us say, right. So what will this particular what do we say rate of or the kinetics of this degradation depend upon now? So I am looking at rate of degradation of this particular compound S. So in the context of biological process, they look at this or refer to this compound as substrate, let us say, right. And again this should be familiar to most people who have had some what we say knowledge of wastewater treatment, let us say, right. Typically they talk about it as substrate, let us say, right. Again this is the compound that is rate limiting or is required for the relevant microbes, let us say, to be able to carry out the relevant degradation, let us say, right. So I am interested in this rate of degradation of this particular substrate or this particular compound. So what is it depend upon? Obviously it depends upon the microbes and the rate at which they are growing, let us say, right. As in typically let us say if you have compound A plus compound B going to products, right, the rate would be R equal to the rate constant times concentration of A, concentration of B and so on, right. Yes, that is if it is a chemical reaction typically that is what we have out there, right. It depends upon the concentration of the relevant reactants and the rate constant, right. But here obviously there are other aspects at play because you have living organisms present. So the rate of growth of these microbes is obviously very much important for to look at or understand the rate of decay or not decay, degradation of this particular relevant compound, right. If your rate of growth of the microbes is higher, typically the rate of growth of the decay or degradation of your relevant contaminant will be higher. As in let us say your contaminant is being eaten up by a few microbes, let us say, Obviously, if they are thriving well and there are more number, let us say, the rate at which this particular contaminant will be eaten up or degraded will be also greater, let us say, right. So, there are different aspects of at play here, and typically we refer to the relevant kinetics as monokinetics, right. So, this is what we are going to look at, right, and again what are the obvious aspects that it's going to or they are going to be dependent upon. So, one case to keep in mind that it's a saturation model, right. It's a saturation model. Again we will look at these aspects. Let us say, keep in mind that there are certain thresholds above which, let us say, or the maximum values above which, let us say, it cannot increase, let us say, right. So, that is going to be the case, as in let us say if I have this particular case R here and substrate concentration here, let us say, right. So, it is going to be, let us say, something like this, let us say, right, and this is my maximum, let us say, R max, let us say, right. So, there is going to be a certain maximum. So, it does not mean as if I have a lot of what do we say food out there for the microbes, they are going to keep increasing and increasing and increasing. There is a maximum rate of growth, right, and again there are other aspects at play. Obviously, what are they going to be dependent upon? They are going to be dependent upon the concentration of the microbes, right, which I am going to represent by X, concentration of the microbes that are present or the amount of microbes that are present. That is going to be dependent upon the amount of food that is present, right. So, again these aspects, and there are other particular what do we say variables out there, and again based on that we are going to come up with an empirical equation which we are going to refer to as monokinetics, I guess, but we are going to look at this in greater detail in the next class. So, for today though I will end this session for now, and thank you.